Welcome back to my course Aspects of Biochemical Engineering. Now, in the last lecture, I started this stoichiometry of biochemical process. In the stoichiometry biochemical process basically deal with the intermolecular relationship of different reactant molecule to give the product. And also I told you the stoichiometry of bioprocess gives the two additional thing. One is it help us to find out the validity of experimental results, and this is on the basis of thermodynamic coefficient of the process, because I told you in the aerobic process the thermodynamic coefficient varies from 0.5 to 0.6, and in case of anaerobic process it is 0.7. So, on the basis of that, now in this lecture I shall show you how we can do the validity of the experiment results, and how we can solve some numerical problems, how we can develop some kind of stoichiometry of the kind of chemical or biochemical processes. Now again let me point out that whenever we do any kind of stoichiometric analysis, I told you first of all we shall have to have the idea of the process. If you don't have the preliminary idea of the process, it will be very difficult to analyze the system. So I hope this will be very clear, because coming to lectures I shall concentrate on solving the different problems. The first thing that I want to point out, this particular problems, the Klebsiella aerogenes is produced from glycerol in aerobic culture with ammonia as nitrogen source. Biomass content 8% of ash, 0.8 gram of biomass is produced for each gram of glycerol consumed. So, no major metabolic products are formed. What is the oxygen requirement for this culture in mass terms? So, basically this this equation deals with that glycerol is used as a carbon source for the production of biomass, and here the biomass that is used, that is the Klebsiella aerogenes, that is the cell mass that is used, and in this process that we get 0.4 grams of biomass is produced per each gram of glycerol forms. Now question comes how we can solve this problem. That let us try this. Now as you know that that yx by s is equal to what? 0.4. Am I right? 0.4 is the yx by s, and what is the glycerol formula? Glycerol is CH2OH, CHOH, CHOH. Am I right? So this is the molecular weight of 92. Now how many carbon atom is there? 1, 2, 3. So per atom, per gram carbon atom, how much will be? What will be the molecular weight of 1 gram atom? This glycerol 93 by 2. So what I can do? This is 0.4 means gram of cells per gram of substrate. Am I right? Now per gram, if I can what I can do, I can divide that with respect to this molecular weight. So this is a cell. We know the molecular weight of the cell. The formula in the previous lecture I have already shown the average formula of the cell mass. So if we take the molecular weight, this is around 26.1 gram, and this is 92 by 3. So this will come around 0.47. 0.47 means that is, this is gram atom biomass or cell, whatever we consider by gram atom of substrate. This we can easily find out. Now why O by S, why O by S is what? That a gram of oxygen required per gram of substrate consumed. It can be written like this. This is gamma S minus Yc into gamma B minus Yp and gamma P divided by 4. Now, now if we here we don't have any product other than biomass, so we can assume this is P is 0 because P we have only biomass is the product. Now, if you put the value here, you will get this is 0.687, and so amount of O2 required, how we can calculate? This is 0.687 multiplied by 3.32 divided by this is 92. This is 92 by 3 and this is 32. So, if you multiply this is coming around 0.72 gram oxygen per gram substrate. So, we can easily calculate. So, let me repeat it again how we have solved this. In this problem I told that Klebsiella aerogenes is produced and this picture is given. This is 0.4 gram of cell produced per gram of substrate. Now, if you want to convert it, that how much gram atom of biomass produced per gram atom of substrate, simple we can divide by the molecular weight, and then we can we have this the oxygen requirement per gram of substrate. This is the formula that we have, that we have already in the last lecture we have seen that. So, if we put the value of gamma S then gamma B here and Yc and divide by 4, we will get this figure. Now, this is actually this is the gram of oxygen required per gram of this is that is moles of oxygen required per gram atom of substrate. Now, if it is like this, what is the molecular weight of oxygen? This is 32. What is the gram atom molecular gram atom 1 gram atom of substrate? 92 by 3. So, if you divide that, we will get 0.72 gram of oxygen that is required per gram of substrate consumed. Now, in this problem, if you go to the text, I hope everything will be clear. Now, here what we have seen, this is the formula of the biomass that we have, if we assume that, and this is how we got the molecular weight, because how we got the molecular weight, this is the actually the molecular weight, but it contains the 8% of ash. So, you divide by 0.92 get the 20. So, 26. So, 100 grams. So, this will be the actual the molecular weight of the this is the total mass of the biomass that we have. Now here that degree of reduction that we can easily calculate like this, and then molecular weight of glycerol I showed you this, and this is the equation that we have. Then Yc, that is already calculated. I showed you this calculation, and this is how we can do it here. This whole calculation we have done it, and this is per gram atom of carbon oxygen requirement. This is the equation that we have. This is moles of oxygen they were produced per gram atom of substrate consumed. The moles the molecular weight of oxygen is 32. And then 72 by 3 is the gram molecular weight of 1 gram atom of substrate. If you multiply, you will get this substrate, that how much oxygen required per gram of substrate. So, now this is how we can do that. Now, next problem that we have, this is little bit different, that following the macroscopic coefficient should determine for cultivation of yeast Candida utilis on ethanol. The substrate consumption of biomass production, oxygen consumption, fermentative heat produced, and then determine the stoichiometry of the process. So, here the question is little bit similar as compared to the previous equation. The reason is that in the previous problem we have considered glycerol as a carbon source now, and organism was Klebsiella, and here we are using the ethanol as a carbon source, and organism is Candida utilis. So, but here we shall have to calculate not only the oxygen requirement, but also we shall have to calculate how much is the heat evolve take place, how much substrate consumption take place, how much what will be the stoichiometry of so complete analysis of the process that is to be taking place in this particular problem. So, now as I told you, as I pointed out, that when you analyze any kind of biochemical process, first we should have the preliminary idea that how this process work. Now here this is the substrate, this is what is the formula of ethanol? Ethanol formula is C2H5OH, am I right? Now this I can write C2H6O. Now if you divide by 2, it is CH3O.5. This I can write. So, this is how it has come here. This is CH3.5. Then oxygen, this is aerobic process, and ammonia required for the cell mass formation, and we get the biomass, and also carbon dioxide and water. This idea we should have what will be the reactant and what will be the product. Then and only then we can analyze the process. Also, otherwise we cannot analyze the process. Now here we can degree of reduction for substrate, biomass, and that carbon atom the ratio, carbon mass ratio, that of the substrate and biomass, that we can easily calculate. Now this is sigma S and sigma B, how you can calculate. This has been shown here. Now, as you know, I told you that aerobic process thermodynamic coefficient varies from 0.5 to 0.6. So, I can assume this is 0.6. Now, if it is 0.6, then I can this is Yx by S equal to this formula we know. So, we put the 0.6, we calculate at 0.92. Now, Yx by O, it can be calculated 3 eta by this. Now, then we can calculate as gram of biomass produced per gram of oxygen used, that we can calculate from this. Here we can calculate. Then we can calculate the oxygen demand. This we have done in the previous equation also. Only the thing is that here it should be Yc, not gamma C. This Yc is this, and eta is this. So we can calculate this. Yc is coming about this 0.856. Now here if you look at this formula, so we are talking about D. Yc means this is here, it will be D. So this is actually Yc. Yc is the gram atom of biomass produced per gram atom of substrate consumed. So we can easily calculate, then from this that is why we have written this is equal to D, and this is the 0.85. Then B we can calculate from this equation. B we find out this, and then we can find out that how much heat evolved. If this is 0.6 we get, and then we 0.6 if you put it is gram atom of carbon substrate, and then I told you this we can convert to gram atom of biomass formation. You simply divide by the yield coefficient. You can do it very easily. And then this is the simple elemental analysis. If you in this equation, if you do the elemental, this is the carbon balance, this is the oxygen balance, this is the hydrogen balance, this is the nitrogen balance. If you do, you will get all the coefficient value. C value, we can G value, different coefficient value, and if we put this stoichiometric equation, we will find this equation is CH3.602, and this you can find out. So this will give you some idea that what is the theoretical yield of the biomass from gram atom of substrate, that you can easily calculate. So you have to, what I suggest you, that you should practice it. Until unless you practice, it will not be clear to you, because it is simple. This material analysis of the process. If you do the material analysis of the process, you can do it very nicely. Now, as I pointed out, that that stoichiometry of the bioprocess also give you the idea that about the validity of the experimental results. Now, this particular problem deals with that. Now, what is this problem? Problem is that during the anaerobic microbial production of ethanol on complex pongolus media containing the sugar, the following experimental data were recorded from a batch culture process, whose concentration is this. This is, so here I want to point out like this, that ethanol fermentation this is considered as the anaerobic process. Now, I have already I explained that the anaerobic process means organism doesn't require molecular oxygen for their growth and metabolism. Not only that, in case of obligatory anaerobe, this molecular oxygen will be harmful for them. Now, this ethanol fermentation process that we carried out with the help of Saccharomyces cerevisiae. Now, present problem deals with that. So, it is, suppose this is a reactor. This is the reactor. In this reactor, we put your substrate and put your cell mass, and under anaerobic condition you run this process, and what will be your product? Your glucose will be converted to ethanol, and also it converted to part of the cell mass. But one thing I want to point out here, that since it is the anaerobic process, the amount of cell mass production will be very less, because in case of aerobic process, if it is aerobic, then cell mass production will be much higher, ethanol will not be there. So, only under anaerobic condition ethanol will be produced. So, what in this present problem, what we have done, we have carried out this experiment. We draw the sample here and we prepared a table. And this is with respect to time. We find out the substrate concentration, we find out the product concentration, we find out the cell mass concentration, we find out that. So, we at different times we can find out the different this concentration. Now, from that we can suppose this is the 0, then we have slowly you come to the maybe your 14 hours, I do not know what is the so initially you have some substrate concentration. So, then finally the substrate concentration usually should be 0, and product concentration usually 0 initially, then finally you have some substrate is converted to products. Cell mass, you have small amount of cell mass concentration which is present as the inoculum, and finally this will little bit increase. These are the things we have. Now, here we can easily calculate what is the x by s value. How you can calculate? If you know that what is the final x value, and we know the initial x0 value, and we know S0 minus S value. S0 is the initial substrate concentration, S is the final substrate concentration here. So, if we can find out easily that what is the biomass yield. Similarly, I can calculate y p by S, that also we can calculate. How we can calculate? This is p minus p0 divided by S0 minus Yes. So I can easily calculate the this is the product yield. So this is how this formula looks. This is how this you see that how how this problem, this is different time, the 0 to 2 hours, 4 hours, 6 hours, 8 hours, 10 hours and 12 hours as the biomass concentration keep on rising. This is initially it is 1 gram per liter and finally it is 6.4 gram per liter. Sugar concentration was 50 gram per liter and finally it is 0, and ethanol 0.04 very trace amount and finally it is 27 grams per liter. So this is the question that we have, and we shall have to find out whether these experimental results, the data that is reporting in this table, whether it is right or wrong. This we shall have to find out. Let us see how we can find out how that validity of the experimental results. Now, what we can do first, that this is the substrate, this the glucose is converted. The glucose, if we consider per gram atom carbon, this is CH2O, you know that. Now, ethanol also C2H6O, this we can write CH3O5, and biomass composition, this average biomass composition is reported, this we can take into account. So, so now from the experimental results we can find out. From the experimental results, we can find out what is the value of Yx by S, what is the Yp by S. This is the initial cell mass concentration. You can find out this. You see that this is initial cell mass concentration. This is the final cell mass concentration. Initial sugar concentration, final sugar concentration, initial ethanol concentration, final ethanol concentration. So here, if you put this data, we will get this two picture. One is that cell mass yield 0.106 gram of cell per gram of substrate consumed. Yp by S is 1.539 gram of ethanol per gram of substrate consumed. So this I can then we can calculate from this empirical formula. From this gram atom of carbon, we can easily calculate what is the gamma S value, what is the gamma P value, gamma B value we can calculate. Similarly, sigma S, sigma B and sigma P we can easily calculate, that we can do the calculation. This is the value we obtained, and then this is Yx by S equal to what? This is the equation we have seen previously. Now, what we shall have to do? We shall have to put all the values. This is the experimental results, and this is the degree of reduction of substrate and carbon fraction of the substrate, and this is the carbon fraction of the biomass and degree of reduction of the biomass. Then if you put all these values here, you can find out the eta value. What is the eta value you get? It is 0.135, and then Yp by S I can write, that is epsilon P equal to gamma S sigma S by gamma P by sigma P. So again, if we this value we have already calculated, and this value already we also calculated from the empirical formula. And so if we put all these values, then we find this is 1.055. Now, so what we know that thermodynamic what is the thermodynamic coefficient? What is the thermodynamic coefficient? This is equal to eta plus epsilon P. So, so what is the eta value? This is the eta value and this is the epsilon. So, it is coming what? This is 0.135 plus 1.055. So, we know that the anaerobic process, the thermodynamic coefficient is how much? Thermodynamic is about, this is about equal to 0.7. So, it is much higher than 0.7, am I right? So it is 1.13191888. That is, it is quite high value. So, so it is clearly indicate the data that is reported here, that is not proper. There is some mistake in the data, because this is not following that the rule that thermodynamic coefficient that we have for the anaerobic process. So, what we have done, this is exactly here it is pointed out, this is 1.19, which is more than 0.7. So, for validation of the data of thermodynamic coefficient anaerobic process should be done. Now, how we can validate that, what is this, we shall have to find out where the we might have committed the mistake. Now in the anaerobic digestion process, what we have what we estimated the glucose concentration, we estimated the cell mass concentration, we estimated the ethanol concentration. Now usually cell mass concentration estimated with the help of gravimetric analysis, it does not have much of mistake. And say the glucose estimation process we do colorimetrically, and in case of the ethanol, we usually do with the help of a gas chromatograph with very microscopic, because gas chromatograph results are very reliable, because it minutely estimate the ethanol concentration. So, what we can expect that out of this data, we can expect the colorimetric analysis of the substrate might be wrong. So, what we have done, we multiplied some factor, because with the substrate concentration, and try to find out that what is the respective value of y p by s and y p by s and y x by s. And what we have observed, if we multiply it by substrate concentration by 1.7, then y p by s value is coming about 0.317 and y x by s 0.062. So, so then if we calculate the eta value and epsilon p value is coming 0.079 and epsilon p 0.621. If you sum do the two things, is coming about 0.7. So, but then and only then that results will be okay. So, from that we can conclude that there is something wrong in the substrate estimation, and if we multiply this substrate concentration by 1.7, then and only then your results will be okay. This is how we can find out the validity of the experimental results. So, in this lecture I try to talk about how we can do the analysis of the biochemical process. I have given two examples. One is with respect to cell mass formation using the aerobic process, and there is the anaerobic process. The aerobic process, we convert the carbon to cell mass, and the anaerobic process we convert carbon to cell mass as well as the product. So, and finally we try to find out how this stoichiometric analysis help us to find out the validity of the experiment results, and I think from that you will get the idea that how this stoichiometric equation can be applied. Also, we calculated the heat evolved for the production of per gram of biomass, per gram of substrate consumed, that also can be easily estimated. Thank you very much.